Hello and welcome. Have you ever stopped to think about the sheer amount of data hiding inside you right this very second? Seriously, modern healthcare is going through an absolute data explosion right now. We're no longer just looking at outward symptoms. We are zooming way in to look at billions of microscopic data points. So the goal of this explainer is to demystify exactly how we translate our genetic blueprints into the active physical cellular machinery that actually runs our bodies day to day. Basically, we're going to decode the big data of life.
Okay, let's dive right in. Think of this as our syllabus for the day, pulled directly from Professor Samith E. Hassanain's fascinating lecture materials on data science in healthcare. We'll kick things off with the raw data, move into the heavyweight bout of genomics versus proteomics, uncover how a single gene can build multiple things, map out the protein pipeline, break down the high-tech tools scientists are using, and finally, look at what all this means for the future of biology.
All right, Section 1, the blueprint and the data. Before we can even begin to understand the incredibly complex system that is the human body, we really need to get a grip on the raw data. To do that, let's build a super simple foundation. Imagine your DNA is this master blueprint locked away in a highly secure vault deep inside your cells. It holds every single instruction for your body. But, the vault can't actually build anything itself. That's where RNA comes in. Think of RNA as the messenger who sneaks in, takes a photocopy of those instructions, and runs it out to the construction site.
Now, as our source material explains, when scientists try to read these biological photocopies, they end up with the literal mountain of digital information. And I know, looking at this, the terminology seems incredibly dense. FASTA, FASTQ, tuxedo suites. But let's completely simplify it. These are really just the foundational computational workflows and digital tools that researchers use to read and assemble our genetic code. Think of FASTA and FASTQ as just specific types of text files that hold biological code, kind of like how a Word document holds text. FASTQC, that's simply a spelling and grammar checker to make sure the data we pulled isn't corrupted. And those brilliantly named tuxedo suites are just software tools used to reconstruct the message, basically stitching those photocopied pages back together into a readable manual.
But here's the catch. Reading billions of microscopic files is never perfect. Our text note's a massive headache for researchers, constantly battling two very confusing signals in their data, which are technical noise versus biological variance. Imagine you're trying to tune into a radio station. Technical noise is just the static caused by a bad antenna or a glitch in the machine. Biological variance, though, is a real change in the actual song being played. It's a true biological difference. So, to tell the difference between a random machine glitch and a real, important biological change, scientists use what are called biological replicates. That basically means they run the exact same experiment multiple times to build an accurate statistical model. If a weird blip only happens once, well, it's just static. But if it happens every single time, that's the music.
Moving on to Section 2, genomics versus proteomics. So we've got the blueprint, but that introduces our next big hurdle. Just having the genetic instructions doesn't actually tell us what's currently being built on the construction site. And that brings us to a massively important word you need to know, the proteome. Coined by a scientist named M. Wilkins back in 1994, this term is essentially just a mashup of the words protein and genome. For the absolute beginners out there, it's important to know that proteins aren't just, a chicken breast or a post-workout shake. In biology, proteins are the actual physical building blocks and the microscopic machines running literally everything in your body. Your hair? Protein. The enzymes digesting your lunch? Proteins. The antibodies fighting off your cold? You guessed it, proteins. The proteome is the complete set of all these proteins produced by an organism. And crucially, it's not static. It changes constantly depending on the time of day or how much stress the organism is under. Which leads us to a really brilliant way to understand the difference between these two massive fields of study. Genomics tells us capacity, like what kinds of proteins an organism can produce. Proteomics tells us action, what roles those proteins are actually playing right this second. Let's use a restaurant analogy because I think it makes it crystal clear. Genomics is like walking into a restaurant and reading the master recipe book. It tells you everything the kitchen could possibly make. But proteomics? That's walking out into the dining room at 7 p.m. on a frantic Friday night, looking at the actual plates on the tables to see what is actually being made, eaten, and maybe dropped on the floor under the stress of the dinner rush. Proteomics gives us a much clearer, real-time picture of reality.
Section 3. One gene, many proteins. For a long time, there was a really common misconception, actually, scratch that, a lot of people still think this today, that biology is a simple one-to-one system. meaning one gene in your DNA creates exactly one specific protein. But biology is infinitely more clever than that. So, what does this actually look like? Well, our source text points to this massive number. According to the Nature 2001 International Human Genome Sequencing Consortium, an estimated 40 to 60% of human genes undergo something called alternative splicing. To put that into a relatable analogy, think of a gene like one massive, incredibly complex Lego set. Instead of just following the instructions to build exactly one spaceship, your body uses that exact same box of Legos to build a spaceship, a car, or a castle simply by combining the pieces differently. One single genetic recipe can yield a whole bunch of different physical proteins.
And the customization doesn't even stop once the protein is built. These molecules undergo intense modifications after they're created. Our lecture materials list things like proteolytic cleavage, where proteins are literally chopped up into new shapes, and these really complex covalent modifications like phosphorylation, methylation, or sulfation. Now don't worry, you absolutely do not need to memorize these names. What you should take away from this is just how heavily customized these proteins get after they leave the factory line. It absolutely proves that biology isn't just a static, boring blueprint. It is a highly dynamic, constantly adapting system.
All right, Section 4, the protein pipeline. With all this mind-bending complexity happening, how do scientists actually track it all? Let's transition into how researchers map out this incredible journey from a simple string of genetic code to a fully working biological machine.
Let's walk through how this builds step by step. This is essentially the central dogma of biology. Step 1, we start with the raw DNA sequence, which is split into individual genes. Step 2, that code gets translated into an amino acid sequence. You can picture amino acids as little beads on a string. Step 3, those beads are linked together to form a basic protein. Step 4, that string then folds into a highly specific 3D structure. And finally, Step 5, that specific physical shape dictates the exact protein function. If the shape is even slightly wrong, the machine just doesn't work. So we're going from raw information all the way down to a physical 3D tool.
And just to show you how quirky and detailed this microscopic world gets, the lecture highlights a really memorable trivia fact regarding those amino acid beads we just talked about. Out of all the standard amino acids used to build life, there's one called selenocysteine, and it's the only one that contains a rare selenium atom. It's a tiny, highly specialized piece of micro-level science, sure, but it really highlights just how unbelievably specific the elemental building blocks of our bodies are.
Which brings us to Section 5, Mass Spectrometry Explained. All this tiny detail begs a massive rhetorical question. If proteins are this incredibly complex, and they operate on a literal atomic level, how on earth do data scientists actually see and measure them?
The answer is a piece of technology called mass spectrometry. Take a look at the sharp, vivid vertical spikes running along the bottom axis of this chart. That bottom line is the m/z axis, which stands for the mass to charge ratio. As our source explains, mass spectrometry basically turns molecules into gas, zaps them with an electrical charge, and then measures them. Think of it as an unbelievably high-tech coin sorting machine. But instead of sorting pennies and quarters by their physical size, it sorts invisible biological particles by their exact weight and magnetic charge. Those spikes tell scientists the precise elemental makeup of a sample, making it the absolute ultimate tool for proteomics. Finally, Section 6, the future of biology. We spent a lot of time today zoomed way in on the microscopic level, looking at RNA files and molecular weights. Let's shift our focus to the big picture and bring all this data back to why it actually matters for us in the real world. This perfectly illustrates where modern healthcare is heading. Biology is changing at lightning speed. We now rely on high-throughput technology, which is really just a fancy way of saying we have machines that can process millions of samples in a fraction of the time it used to take. That leads to massive, exponential increases in data, and entirely new types of data. But the most absolutely crucial point here is that last one, a major emphasis on systems biology. We aren't just looking at a single cell or a single gene in isolation anymore. We're using massive computing power to understand how the entire system interacts, all at once.
If you let your eyes trace this data pipeline from left to right, you can really see how it all connects. Over on the far left, you have all the tiny stuff we've been talking about: nucleotides, proteins, cellular pathways. Right in the middle, we integrate all that microscopic data into the whole human being. And then, as we move to the right, we scale up to entire human populations.
Our source makes it clear that this integrated data pipeline translates directly into real-world life-saving applications: biobanks, new targeted medicines, disease prevention, and catching illnesses far earlier than ever before through advanced early diagnosis.
Systems biology, powered by proteomics and all this incredible data science, is truly our greatest modern healthcare tool. By finally understanding both the recipe book and the exact meals being cooked on our cells, we are basically unlocking the ultimate cheat codes to the human body. So I want to leave you with this provocative thought. If data science can successfully read our genetic blueprint and track every single microscopic cellular machine we build, what previously incurable diseases will we conquer next? The future of humanity is written right there in our data, and we are finally learning how to read it. Thanks so much for learning with me today.